Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we are going to start a new unit, the renal physiology. Let us begin with the first chapter of this unit, urine formation by the kidneys. First, let us have a brief look at the functions of the kidney. Kidneys perform various functions in your body, but among these, the most important and foremost function of the kidney is to remove metabolic wastes and foreign chemicals from the body. As you already know that as a result of metabolisms of all the cells of your body, certain waste products are produced. These waste products are removed from the body in two forms. Either they are metabolized by the liver and removed from the body in the form of feces, or they are removed in the form of urine with the help of kidneys. Certain drugs are also removed by the kidneys from your circulatory system. Secondly, the kidneys are involved with the regulation of water and also in electrolyte balance. In the coming chapters, we are going to study how various electrolytes are balanced within the body, especially sodium and hydrogen ions and potassium ions. You have already studied how water is regulated within the body. You might be familiar with the role of ADH and with the role of aldosterone. Either way, we are still going to discuss them in detail in further lectures of this unit. Kidneys are also involved in the regulation of arterial pressure. As you might remember from our previous discussions in the unit of kidney, while discussing the topic of long-term control of blood pressure, we discussed in detail how the kidneys activated renin-angiotensin and then renin-angiotensin-aldosterone systems to maintain blood pressure and to regulate blood pressure. The kidneys are also involved in acid-base balance, and how they do this, we will be discussing that quite in detail in our lectures later on. The kidneys are also involved in secretion, metabolism and excretion of hormones. For secretion of hormone, you must remember that a special hormone known as erythropoietin is secreted from the peritubular interstitial cells of the kidney. Then there is secretion of renin by the JG cells of the kidney. Please do not confuse the sites of formation of both of these hormones. They are often asked of you in the MCQs, and students tend to confuse the sites of production of erythropoietin and renin. So many hormones are also metabolized in kidney, such as vitamin D, and almost all of the hormones which are secreted by various glands in the body after completing their functions are removed from the circulation with the help of kidney and excreted from the body in the form of urine. As I mentioned already that kidneys are involved in the production of RBCs, which is known as erythropoiesis, and around 90% of the erythropoietin is produced by the fibroblast like peritubular interstitial cells of the kidney, and rest of the 10% is produced by the liver. So these are some MCQ nuggets. Then kidney is also involved in gluconeogenesis. It might come as a shock to you or as new information to you that kidneys are involved in the process of gluconeogenesis. Now, gluconeogenesis is the formation of glucose or synthesis of glucose from metabolites such as amino acids, and kidneys are in fact involved in the process of gluconeogenesis, and around 10% of total gluconeogenesis occurring in the body happens here in the kidneys, and the rest 90% takes place in the liver. So these are the functions of the kidney. So let us now discuss the physiological anatomy of the kidneys in detail. So the kidneys lie on the posterior abdominal wall and they lie outside the peritoneal cavity. Now what they mean is, let me get this. Suppose if this is an upright person and this is the anterior side and this is the posterior side, the kidneys are attached to the posterior wall of the abdomen. And as I mentioned before, that all the organs of the abdomen are present within the peritoneal cavity and are covered by a special lining which is known as peritoneum. But the kidneys they lie outside the peritoneal cavity, and the peritoneum is reflected on the anterior surface of the kidney, and it does not encase the kidneys. So I hope you understand that the kidneys lie outside the peritoneal cavities, and this is the peritoneal cavity, and here you can see the kidney lying outside the peritoneal cavity attached to the posterior wall of the abdomen. It is in fact anteriorly covered by the peritoneum, and this is how it is present within your abdominal cavity. The kidneys weigh 150 grams each. They are the size of your clenched fist, and they have a structure like that of a bean, and there is an indentation on the medial edge, and this is known as the hilum. And it is from the hilum where the lymphatics and the ureter and arteries and veins which are entering and exiting the kidney, they pass through the hilum or originate through the hilum. And you can also see a ureter coming out of the hilum, and ureter is a tube which carries the urine from the kidney to the bladder, and it is the urine is stored within the bladder until it is emptied during micturition. The kidneys are surrounded by a tough fibrous covering which is known as the capsule of the kidney. So let us have a look at the organization of the urinary system. Here you can see two kidneys lying within the abdominal cavity. From the kidneys arise two tubes known as the ureters, and they are made up of smooth muscle cells, and they carry the urine from the kidneys towards the bladder. Here you can see the bladder. It is also made up of special type of smooth muscle cells which are known as detrusor muscle cells, and bladder is involved with the storage of urine until it is emptied during micturition, and the bladder is emptied into the exterior through a tube known as the urethra, which can be seen over here. Let us now have a look at the internal structure of the kidney. Here you can see the indentation which I mentioned before. This is known as hilum. It is from here where the renal artery enters, the renal vein exits, and the lymphatic vessels also enter through the hilum, and as you can see the ureter exits through the hilum. So the kidney, when it is sectioned, you can majorly divide it into two parts. The outer portion is known as the cortex, and the inner portion is known as the renal medulla, and these two are separated in this figure by this dashed or uncontinuous line which you can see throughout. So the outer portion of the kidney is known as the cortex, and the inner portion is known as the medulla. The medulla is divided into pyramidal shapes, and they are rightly named the pyramids, and you can see them over here. The base of the pyramids lies at the junction of cortex and medulla, while the apex lies towards the hilum. And the apex is known as papilla, and it empties into a extension of the ureter, which is known as the pelvis. The pelvis is divided like a stag's horn. As you know that stags have horns which are branched and divided. So similarly, the renal pelvis, when it moves towards the cortex of the kidney, it is divided into stag horns, and the larger divisions are known as major calyces. Here you can see the major calyces, and the further smaller branches or divisions or the terminal branches are known as minor calyces. So the papilla of the pyramids, they drain into the minor calyces. The minor calyces join together to form major calyces, and the major calyces join together to form the pelvis. And the pelvis then narrows down and exits the kidney in the form of ureter. So let us discuss this in detail. As I mentioned before, the kidney, when bisected, can be visualized and differentiated into two major regions, the outer cortex and the inner medulla. So here you can see this outer region is the cortex, and this inner region is the medulla, and I have differentiated it with this dotted line over here. So the portion which lies outside is known as the cortex of the kidney, and the portion which lies towards the center is known as the medulla or medullary cortex, medullary region of the kidney. And here you can see the hilum, and as I mentioned before, the ureter exits through the hilum, and the renal artery enters, and renal vein exits through the hilum. The medullary let us now discuss them in detail. The medulla is divided into 8 to 10 cone shaped pyramids. Here you can see them. These are the pyramids. They are actually shaped like pyramids. That is why they are known by this name. So these pyramids are so placed that their bases lie towards the medullary corticomedullary junction, where the cortex and the medulla meet, and their apices lie towards the center of the kidney, and this apex is known as papilla. You can see over here that these apices are known as papillae, or singularly papilla. These papilla project into the renal pelvis. So here you can see the pelvis, and the papilla, they open up into the renal pelvis. The renal pelvis is actually the funnel shaped upper broad end of a ureter. This is the ureter, and this is the upper broad funnel shaped end of the ureter. It is divided into major and minor calyces. As I mentioned before, the pelvis is divided like a stag's horns, and the larger divisions are the major calyces, which further divide into minor calyces. So the urine which is formed in the medulla is secreted into the minor calyces by papillae, and from the minor calyces it moves into the major calyces, and from there to the pelvis, and ultimately into the ureters. Another interesting information is that the walls of these major and minor calyces, along with walls of pelvis and ureter, are contractile in nature. They contain contractile elements. Why? Because they are made up of smooth muscle cells, and these smooth muscle cells have actin and myosin within them, which allows them to contract. And this is very helpful to propel the urine from the kidneys towards the bladder, where the urine is stored until it is emptied during micturition. Let us now discuss the renal blood supply. Do you know that around 22% of your cardiac output is supplied to your kidneys? What is your cardiac output? It is the amount of blood which is pumped by the heart per minute, and it is around 5 liters or 5000 ml. Out of this 5000 ml per minute, 22% is being delivered to the kidneys. This amounts to about 1100 ml per minute. And as you can see that the kidneys receive very large quantities of blood as compared to their size. Their size is really small, but the blood supply they receive per minute is huge, and the reason for this is that because they play a very important role in filtering the blood and in removing waste products of the blood. So they it is important that they receive large quantities of blood per minute, so more blood can be cleared of waste products and filtered and returned to the circulation. So let us now discuss the renal blood supply. Let us first discuss the arrangement of blood vessels within the kidney. As you can see that the renal artery, which is a branch of aorta, it enters the kidney at the hilum. So this is the first artery which is entering the kidney, and this is known as the renal artery. It then divides into interlobar arteries. What are interlobar arteries? Basically, these are the branches of renal arteries which are present between the pyramids. You can see the pyramids over here. This is one pyramid, this is the second, third, fourth, fifth. So the branches of the renal artery which divide between the pyramids are known as interlobar arteries. These interlobar arteries arc at the base of the pyramid and form this arch like shape, and that is why they are known as arcuate arteries. You can see over here that the interlobar arteries are arcing at the base of the pyramid. That is why they are known as arcuate arteries. These arcuate arteries give off smaller branches like hair, and you can see them over here. These branches are known as interlobular arteries. Do not confuse them with the interlobar arteries. These smaller branches which originate from the arcuate arteries are known as interlobular arteries. So these interlobular arteries, you can see them over here. This is an interlobular artery. These interlobular arteries give off efferent arterioles, and you can see them over here. These efferent arterioles form a capillary bed which is known as glomerulus. You can see the glomerulus over here. This is very important structure and very important part of the nephron. It is here where all the filtration takes place. Then unlike other parts of the body, as you already know, in most part of the body there are efferent arterioles, then capillaries, and then these capillaries form together to join together to form venules which form veins. But the capillary bed within the kidney is different. This capillary bed is basically joined by two arteries. In fact, your kidney has two capillary beds. So the arrangement is efferent arterioles, they divide to form a bed of capillaries which is known as glomerulus. These capillaries join together to form efferent arterioles, and you can see the efferent arterioles over here. Then these efferent arterioles again break up to form another group of capillaries, another bed of capillaries, which are known as peritubular capillaries, and they are present around the loop of Henle and other parts of the nephron. These peritubular capillaries then join to form the interlobular vein, and you can see it over here. These are the peritubular capillaries which join to form the interlobular vein, and you can see the interlobular vein over here. The interlobular vein then forms arcuate vein, and the arcuate vein forms the interlobar vein, and the interlobar veins join to form the renal vein. So this is the organization of blood vessels within the kidney. Here is another representation of the same concept. Here you can see the renal artery which enters the kidney at the hilum. This renal artery forms large arteries known as segmental arteries, and these are the branches of renal arteries. Segmental arteries give off branches between the pyramids, and these are known as interlobar arteries. Interlobar arteries arcuate at the base of the pyramid and form arcuate arteries. Arcuate arteries give small hair-like projections. These are known as interlobular arteries. Here you can see a zoomed version of the interlobular artery. The interlobular artery gives off the efferent arteriole. Efferent arteriole divides into or branches into capillaries, and this capillary bed is known as glomerulus. Then the glomerular capillaries join to form another artery which is known as efferent arteriole. So as I mentioned before, this is the difference between the capillary bed of the kidney and the capillary bed in elsewhere in the body, where capillaries have arterioles at one ends and venules at other ends. This is now not how it happens in the kidney, because in kidney there are present two capillary beds, a glomerular capillary bed and a peritubular capillary bed, and they are joined together by efferent arterioles. So the interlobular artery divides into efferent arteriole, which divides into the glomerulus, which is a tuft of capillaries. These capillaries join to form efferent arterioles. Efferent arterioles again break up into capillaries which are known as peritubular capillaries, which surround the nephron, and these capillaries then join to form interlobular veins, which join to form the arcuate veins, and these arcuate veins drain into interlobar veins which are present between the pyramids, and these interlobar veins drain into segmental veins which join to form the renal vein. So this is how the arteries within the kidney are arranged. So you might ask, what is the purpose of two capillary beds within the kidney? This is very important. The first capillary bed is responsible for filtration of fluid. So it has very high pressure. Fluid is filtered out of the glomerulus into the nephron, and along the nephron are present the peritubular capillaries, and these capillaries are responsible for reabsorption, and most of the fluid which is filtered at the glomerular capillaries is reabsorbed at the peritubular capillaries, and these peritubular capillaries are present in close proximity of the nephron. You can see जहाँ पर loop of Hamle नीचे जा रहा है, peritubular capillaries भी नीचे जा रही हैं, and the function of peritubular capillaries is reabsorption of most of the fluids and nutrients such as glucose and amino acids, and most of sodium and also other ions. And peritubular capillaries, they pick up all of the substances which are required by the body, which were filtered at the glomerulus, and carries these important substances back to the circulation. So remember, glomerulus is responsible for filtration of everything, and peritubular capillaries are responsible for reabsorption of important nutrients. So to sum it up, your kidney has a unique circulation because it has two capillary beds, and these are the glomerular capillaries and the peritubular capillaries, and you can see them here in the figure. I have already explained to you what is their arrangement and what is their function. So an important point is that by adjusting the resistance within the afferent arteriole and the efferent arteriole, you can control or alter the hydrostatic pressure within the glomerular capillaries. And by controlling the hydrostatic pressure in the glomerular capillaries, you can control the filtration which takes place from the glomerulus into the Bowman's capsule. Similarly, you can also control the reabsorption of fluid in the peritubular capillaries by readjusting the resistance within the afferent and efferent arterioles. How this happens and what is the effect of increasing or decreasing the resistance of afferent and efferent arterioles, what is its effect on hydrostatic pressure in the capillary beds, we will be discussing that later on in greater detail. So here is a really simple, colorful and easy figure or diagram to understand the circulation within the kidney and the function of the capillary beds within the kidney. So here you can see the blood which is pumped by the heart. It is entering into the kidneys, and this blood passes through different branches of renal artery until the branches narrow down or form small afferent arterioles. And here you can see the afferent arteriole. Remember that afferent arterioles contain the blood with all the waste products and metabolites from your body. So this blood vessels, which are the afferent arterioles, break down into a bed of capillaries which is known as glomerulus, and the pressure within the glomerulus is so high that a lot of fluid along with nutrients and ions is filtrated from the glomerulus into the Bowman's capsule. So this filtrate travels along the parts of the nephron from the Bowman's capsule to the proximal convoluted tubule, to the loop of Henle, to the distal convoluted tubule. And coming back to the capillaries, the glomerular capillaries then join to form another arteriole which is known as the efferent arteriole. This efferent arterioles again breaks up into a capillary bed which is known as the peritubular capillaries. These peritubular capillaries are present all around the nephrons, especially the proximal convoluted tubule, the loop of Henle and the distal convoluted tubule. What these blood vessels do is, what this group of capillaries, the peritubular capillaries do is, they pick up almost all of the fluid from the nephron. They pick up almost all of the nutrients such as glucose and amino acids which were filtrated at the glomerulus. They reabsorb them, and they also reabsorb majority of ions which are required by the body, and they do not, however, pick up the waste products. The waste products remain within the nephron until they are excreted in the form of urine. So what is left behind is filtered pure blood which is reabsorbed by the peritubular capillaries, and these peritubular capillaries carry them to the general circulation in the form of veins. This is how blood is purified within the nephron. We will be discussing this concept in detail later on, but I hope you got the general idea. Let us now discuss the structure of nephron. So nephron is basically the structural and functional unit of your kidney, and each of your human kidney contains about 800,000 to 1 million nephron, and each of this nephron is capable of forming unit. So let us discuss the parts of nephron. First of all, we have the glomerulus. Glomerulus is not actually structural part of the nephron. It is in fact a functional part of the nephron, because you already know that glomerulus is a tuft of capillaries which is in fact a part of the circulatory system of the renal medulla. It is not in fact the part of the nephron, but it is a very important functional part of nephron, so that is why we include it as a part of the nephron. So actually the part of the nephrons include the Bowman's capsule and the renal tubules. So among the renal tubules we have the proximal convoluted tubule, the loop of Henle, both the descending and the ascending limbs. Then we have the distal convoluted tubule and the collecting ducts and tubules. So these are the parts of nephron. You can see them over here. Here we have the glomerulus. As I mentioned before, it is the structural part of the circulatory system of the kidney or the blood vessels of the kidney, but it is a functional part of the nephron. Because without glomerulus there is not going to be filtration and there is no use of the tubular part of the nephron. So it is a functional part of the nephron. And as for the nephron, it consists of the Bowman's capsule. You can see it over here. It is present around the glomerulus. Then you have the distal convoluted tubule, which then forms the loop of Henle. There are two limbs of loop of Henle, the descending limb and the ascending limb. The ascending limb is made up of two portions, the thin ascending limb and the thick ascending limb. The thick ascending limb then continues as the distal convoluted tubule. The distal convoluted tubule then forms the collecting tubules, and collecting tubules join together to form the collecting duct. So this is how the nephron is actually placed. I also told you about this special arrangement of the nephron in circulation, in the unit of circulation when we discussed the renin-angiotensin system. And let me clear it again that normally you see this kind of structure of nephron within your books. This is not how actually the nephron is placed within your kidney. This is just a schematic diagram to help you understand the different parts of the nephron and different functions of these different parts of the nephron. Actually in your kidneys, this is how your nephron is placed. Your distal convoluted tubule must always pass between your afferent and efferent arterioles. This is how your nephrons are arranged actually within your kidney. So ये जो आपको खोल कर दिखाया जाता है ये सिर्फ आपकी अंडरस्टैंडिंग के लिए दिखाया जाता है. असल में नेफ्रॉन अपने ऊपर ट्विस्ट हुआ होता है और जो आपको ये बुक के अंदर ये पार्ट दिखाया जाता है एक्चुअली जो डिस्टल कॉन्वोल्यूटेड ट्यूब्यूल है इट मस्ट पास बिटवीन द एफरेंट एंड इफरेंट आर्ट्रियोल्स टू डिटेक्ट द कंसंट्रेशन ऑफ सोडियम एंड रेगुलेट ब्लड प्रेशर. Also your glomerulus and distal and proximal convoluted tubules are present within the cortex of the kidney, and the loop of Henle is dips down into the medulla, and this arrangement is very important for formation of concentrated urine. There are two types of nephrons, cortical nephrons and juxtamedullary nephrons, JG nephrons. JG nephrons are so arranged that their loop of Henle's are very long and they dip down deeper into the medulla, and they are responsible for forming concentrated urine, while the cortical nephrons are small and they are present mainly in the cortex, and their loops of Henle do not dip down deep into the medulla. So the regional differences in the structure of nephron, let us discuss these regional differences. There are two types of nephrons on the basis of regional regions within the kidney, the cortical and juxtamedullary nephrons. The cortical nephrons are small and their glomeruli and Bowman's capsule are present within the outer cortex, as I already showed over here. You can see that this is a cortical nephron, and it's most of its portion, not only its glomerulus and Bowman's capsule, but most part of its loops are also present within the cortex or outer portion of the kidney. So another important distinction is that they have very short loops of Henle which do not dip down deep into the medulla, and entire tubular system is surrounded by extensive network of peritubular capillaries. We have already mentioned that peritubular capillaries are very important. They help in reabsorption. Second type of nephrons that we have are the juxtamedullary nephrons. They make up around 20 to 30% of your total nephrons. They lie deep within the renal cortex and renal medullas. They have very long loop of Henles which dip down deep into the medulla, and their peritubular capillaries have a special name. It is known as vasa recta. So do not get confused when you hear this special word vasa recta. Vasa recta are basically peritubular capillaries of JG nephrons which have very long loops of Henle. So these special peritubular capillaries are known as vasa recta, and it is your juxtamedullary nephrons which are responsible for formation of concentrated urine. And how they do this, we will discuss this detail in our next chapters. Here again you can see the graphical representation of the cortical nephrons and the medullary nephrons, juxtamedullary nephrons. So from what we have learned till now, you can be asked a question regarding the differences between the cortical nephrons and the JG nephrons. So the first difference is that they are more numerous, and they form around 80 to 90% of total nephrons, while JG nephrons form around 10% or 20% of total nephrons. Your cortical nephrons are small and have small loop of Henles, while your JG nephrons are large and have large loops of Henle. Your cortical nephrons lie majorly in the cortex, especially their glomerulus and their Bowman's capsule and their distal convoluted tubules lie within the cortex, and most part of their loop also lies within the cortex, while major portions of your JG nephrons lie at the JG junction, the corticomedullary junction, and their loops dip down into the medulla. The cortical nephrons have small loops and the JG nephrons have longer loops. And the peritubular capillaries are which are present around the cortical nephrons are small, while the peritubular capillaries which are present around the loop of JG nephrons are longer and are known as vasa recta. So these are the differences between cortical and JG nephrons.